Good morning, guys.
Good morning.
Good morning.
Morning.
How was your break?
It was
Good.
good. How was yours?
Good. I'm still eating turkey. I think I had so much turkey that I will not have any more turkey until next Thanksgiving. So, all right. So, what do we have? We have Monday, November 30th, and this is our last lecture this semester. And it's going to be on chapter 12. We need to finish the second half. And so what do we have? Wednesday, we have a test. And then what I was thinking now, as far as I would like to have some kind of review session for your final so that you know what's going to be on the final more or less. So I would like to have it earlier. So perhaps maybe on Friday, because if I do it during regular class time on Monday, then you don't have much time to study after that. So I was thinking maybe doing it on Friday and like around 9 o'clock, which is the time for my other section. But if you can't attend it, Friday 9 o'clock, you can obviously listen to the recording of it. And also what you can do is you can email me your questions. So if you can't attend the review session on Friday, email me your questions before that, and I will answer them during that review session, and you will just listen to the recording of it. But obviously you're also welcome to email me and ask any questions anytime before the final anyway. So it's up to you. So you have lots of options. Will this work? Yeah?
Yep.
Okay. So also final, keep in mind, final is a comprehensive test, but the types of questions that I put on the final are a little less difficult than what you get during the semester because it's a comprehensive test. So as far as the time, it's going to be 2 hours for 60 questions, so very similar amount of time. And keep in mind, if you do well, I will replace your worst exam by what you get in the final. So in principle, if you haven't really gained that many points up to this point in time, you haven't lost anything. So in other words, out of 2 tests, if one of them is really bad and the other is reasonable, then really you haven't lost anything that cannot be fixed. So but on the other hand, also keep in mind that I'm sure there are some reasons why you guys, some of you did not do well on the tests. So those reasons have to be fixed, right? So to keep hoping that you do well in the final, but not doing anything about it is not going to help you, right? So something fundamentally needs to change in terms of how you study, how much you study. Okay, so you can always talk to me about that, email me, or we can even, if you want, we can even schedule some Zoom meetings. So are there any questions?
No questions, but I think the recording needs to be started.
I'm recording.
Oh, is it? Maybe I just missed it.
No, I think it says recording. Yeah.
How many
okay.
questions? sorry. I'm sorry. How many
But thank you,
questions did you say?
thank you for reminding. But I think it is recording this time. Yes. Yeah. Question.
How many questions did you say was going to be on the exam and on the final?
60.
60 on the final?
Yeah.
Okay.
Do you want more?
No.
Yeah, I looked at on the previous finals, we had 75. You know, I don't really, I think 60 is plenty. I mean, why do we need 15 more, right? So if I think 60 is sufficient for you to show that you know the material, you don't know the material. Adding 15 more just not gonna do, it's really not gonna change things fundamentally. So, yeah. All right, anything else? All right. How many questions for test 3? Same, 30. 30 questions for test 3. So that will be on Wednesday. Don't miss that. Don't miss that. All right.
Oh, are you going to start test 3 early again? Where we can access it a little bit earlier, like last time? Like, I think you did like 5 minutes before class or something like that.
Okay. Yeah, I'll do the same. Yeah. Just for you to kind of, make sure your technology is working, right? Yeah, sure.
What's the time limit on the final?
2 hours.
2 hours. Okay. Thank you.
So as I said, it's 2.5 hours for 75 questions. So it's going to be 2 hours for 60 questions. All right. Let's go then to the material of chapter 12. So we stopped last time. So we talked about GPCRs, right? We talked about the diacylglycerol and the inositol triphosphate. Right, and so let's pick up from here. So as I mentioned to you, that's chapter 12, very little actual biochemistry, well, mostly biology, a lot of abbreviations. But we still have this chapter in the biochemistry textbook because, nowadays there's so little separation between different fields of science. People work on the interface of many different disciplines, lots of collaborations. So it's useful to know. So even, for example, for research in my lab, where we focus primarily on synthetic chemistry, right, putting molecules together, most students still know what diacylglycerol is. Most students still know what protein kinase C does, for example. We still discuss these things, and because it helps. It helps when we get data from collaborators, we know how to interpret it. So, all right, so these are second messengers. Now, what we need to mention is that, so remember when the hormone binds specific receptor, this is a GPCR receptor, there are 2 second messengers, right? There's IP3, inositol triphosphate, and there is diacylglycerol. So IP3 binds to a calcium channel on the ER in the plasmic reticulum, allowing for the calcium to float into the cytoplasm. And normally calcium concentration is kept low in the cytoplasm, but once the calcium gets into the cytoplasm, it it starts doing things, right? It's kind of a signal for the cell to act, to do something. And one of the main proteins which facilitates that is calmodulin. So calmodulin, it modulates the function of many enzymes. So this is the X-ray structure of calmodulin. And in the absence, so it binds to 4 calcium ions. You can see it's 2 spheres here, 2 calciums. And when it finds one of its client proteins, so basically client protein, the protein that calmodulin does work on. And so the client protein will bind to the central helix, making calmodulin bend, change the conformational preference to this bent form, and in some way activate this protein that happens to be trapped inside calmodulin. And now one thing to keep in mind here is there's this EF hand, which is a site in the molecule where the helix binds to there are 2 helices come together and there's a loop. So there's E helix and F helix, and there's a loop between them. And so this is the binding site for calcium. So very very well-known site on calmodulin, known as EF hand. All right. So calcium modulates the function of many enzymes through calmodulin. Yeah, so all these enzymes are going to be regulated by calcium. So obviously I don't want you to memorize these, but use this as reference in case you ever need that. All right, so GPCRs. Some other use of GPCR. So for example, here, GPCR is used in vision and olfaction. So let's look at the vision one. So very similar to what we already saw, just different variation and different types of particular proteins and enzymes. So first of all, there is a molecule. Remember, we talked about cis-retinal. So we talked about that in a lipid context. So this molecule is present as 11-cis form, and when it absorbs light, so this molecule is a polyene, right? It has a highly conjugated system, if you remember, retinal. If you don't, look at the structure again in the lipid chapter when we talked about lipids. So because it's highly conjugated structure, it will absorb light in a visible region, right? Specifically absorb light very strongly between 400 to 700 nanometers, right? Wavelength. So, and it's bound to a protein known as rhodopsin. So when this conformational change occurs, you can see the molecule of retinal is bent because it's a cis form, right? Cis form is bent. Now transform, on the other hand, is linear. Right? So when it goes from bent to linear, that will affect the rhodopsin quite a bit and will make it change its conformation. Will make it change its conformation. And specifically, once that happens, so this is the kind of GPCR receptor. Now the G protein here is the letter letter T is used for transducin. Transducin is the same as the G protein. So here we have three hetero subunits. So alpha, beta, gamma. So beta, gamma dissociate from alpha, and alpha binds to GTP, right? So GDP leaves, and guanosine triphosphate comes in. And so in this form, the alpha form of transducin with its bound guanosine triphosphate will remove the inhibitor from an enzyme known as phosphodiesterase. Phosphodiesterase. We remove the inhibitory subunit, and phosphodiesterase becomes active. Phosphodiesterase becomes active, and it starts breaking down cyclic GMP to 5' GMP monophosphate. Once that happens, cyclic GMP was the role of cyclic GMP was to keep the sodium-potassium channel open, right? Sodium-potassium channel open. Once the cyclic GMP is destroyed to produce the monophosphate, the channel closes. The channel closes, and so the influx of sodium and calcium stops. As the influx of sodium and calcium stops into this rod cell, I should say this is the rod cell, not a cone cell. This is the rod cell of the of our ocular system. The calcium basically stops coming into the cell. The membrane, hold on a second. The membrane becomes hyperpolarized. So we will later on in this lecture, we will see that the membrane is positively charged on the outside and negatively charged on the inside, okay? And so if sodium and potassium stay on the outside and stop coming in, the membrane, the positive charge on the outside of the membrane even becomes larger, right? Because we keep positively charged ions outside, not letting them in. And so the membrane is hyperpolarized, so it's even more polarized, right? So the plus charge even increases. And that's that signal is sensed, and it's passed to the brain through the optical nerve. And but then after a certain period of time, sodium hold on a second. But this ion exchanger, where you have 4 sodiums coming in and 1 calcium going out, continues to be open. It's constantly open, right? And so the calcium will finally get out, and at the expense of 4 sodium ions. So the membrane is no longer hyperpolarized, because what we have 2 pluses going out and 4 pluses going in, right? So the plus on the outside becomes smaller. So the membrane is not so hyperpolarized anymore. And eventually, so this calcium here that was present, its concentration drops, and reduction in calcium concentration then activates the enzyme known as guanylyl cyclase, GC, which converts guanosine triphosphate to cyclic guanosine monophosphate. The signal is transmitted further. Now you can see here rhodopsin was in a sort of bleached form where it was triply phosphorylated with the arrestin. We saw that already before, right? So arrestin basically takes the rhodopsin out of the game, and eventually it the drop in calcium concentration releases rhodopsin, which is then recycled back and ready for the next photon of light. So the mechanism, even though there are different players, right? They're different, but you can see the fundamental is the same mechanism, right? So instead of G protein, it's a T protein, but the same heterotrimeric protein with the alpha subunit, right? And it's a GTPase. It's a GTPase. All right. And then we're ready for another cycle. All right, any questions? Okay, so that should be enough for GPCRs. Let's move to the second main type of receptors, membrane-bound receptors, and those are known as enzyme-linked membrane receptors. So these will have an extracellular ligand binding domain and intracellular catalytic domain. And so the intracellular catalytic domain will have, in most cases, will have tyrosine kinase activity. So I kind of told you about this last time, but it's good to see it again because it's a fundamental process in signal transduction. So you have tyrosine, the enzyme receptor, tyrosine kinase, RTK, and that will phosphorylate the oxygen, phenolic oxygen on the tyrosine. Okay, and so obviously now this creates, so this is phosphotyrosine, which is now accepted by many different proteins as a binding site. Of course, so just think about it. You have one small neutral OH, right? And now you have a large phosphate with two negative charges. So conformational preferences are very different now, and the binding partners are very different now. So in many cases, this kind of phosphorylation will occur through a phenomenon known as autophosphorylation. So just think of it. So the intracellular portion can be big, right? So some of it may have tyrosine kinase activity, and some of it may have tyrosine residues by themselves. And so the protein will phosphorylate itself. It kind of bites back, right? Bites back and phosphorylates itself. And by doing so, it creates a binding site. So other proteins can come in and also undergo phosphorylation, right? And so it will add phosphate group to a tyrosine in specific target proteins. And we'll also look a little bit about, we'll also talk a little bit about guanylyl cyclase activity. Guanylyl cyclase, we just saw that in the vision, right? But specifically, it converts GTP to cyclic GMP second messenger. So let's look at this now. So receptor guanylyl cyclase, abbreviated as GC, guanylyl cyclase, So what it does, it grabs GTP, guanosine triphosphate. Here's G. Here's OH. Oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus, oxygen. Now the lone pair on this oxygen will attack the phosphate, and the pyrophosphate will be the leaving group. To create a molecule, cyclic guanosine monophosphate. Like so. Cyclic guanosine monophosphate. So oftentimes this is referred to as second messenger. Second messenger. And so, so this is implicated in many different functions, many different tissues. For example, ANF, atrium natriuretic factor, which is a protein produced by the heart, when the blood pressure is very high. When the blood pressure is high, right? Basically, it pushes against the wall of the heart, and the heart basically feels the pressure from the blood when the blood pressure is high, and it produces this factor, which travels to the kidney, and binds to the GC, guanylyl cyclase receptors on the surface of kidney cells. What is the main type of kidney cell? Anybody knows?
Nephron?
Nephrons. That's right, nephrons. So this will bind to the GC, guanylyl cyclase, on the surface of nephrons, right? And the signal will be transmitted inside, producing this reaction. So guanosine triphosphate will undergo conversion to cyclic guanosine monophosphate. Okay, and so these are membrane spanning GCs. And so when that happens, this then results in chloride excretion. I forgot how to spell excretion. E-X-C-R-E. Is it S?
T-I-O-N.
T-I-O-N. Excretion. Okay. Sodium chloride excretion. So, and then what happens after salt is basically excreted through urine? What happens to your blood pressure?
Doesn't water follow? And so the blood pressure is lowered?
Yeah, so basically you just decrease the volume of your blood, and so the blood pressure drops. So it's one of the main mechanisms for the reduction in blood pressure is through this signaling through receptor guanylyl cyclase. Now, a similar process also occurs with another type of peptide, which is now produced also in response to high blood pressure. Now I can't remember what produces this one, guanylin. I think I looked it up, but I can't remember now. So anyway, its receptor is on the endothelial cells in the lumen of the gastrointestinal tract, primarily the intestinal system. And what happens here, again, it binds, and what happens next is guanylyl cyclase will produce the cyclic GMP, which will lead to the chloride excretion from the blood into the intestines and out. And so how will that lower the blood pressure? So now salt is reduced through the, not through the urination, but through number 2, right? So the result is diarrhea, right? The result is diarrhea. I want it not to spell this, but I'm gonna try. Is this correct? I think this is correct.
Yes, that's right.
All right. So diarrhea, right? And so, for example, also these kind of receptors, they also interact with bacterial toxins. So, for example, E. coli, different strains of E. coli can produce toxins, which can also activate these guanylyl cyclase receptors in the intestines, right? And that may result in diarrhea. So, for example, you know that, for example, tap water, there's plenty of E. coli in tap water, right? A lot of it is good. So it's good for us, symbiotic relationship, but a lot of it produces endotoxins, which are not good for us. Maybe good for some people, but not for others, right? So people's bodies are different in terms of what kind of molecules produced by E. coli are accepted and which ones are not. And so oftentimes you drink your tap water at home, you have no problem. But then you go to visit somebody and you try tap water there, and then you have diarrhea. So so that comes from the different strain of E. coli producing different endotoxins. And but the reason for that is this guanylyl cyclase signaling, which produces cyclic GMP and a reduction basically in excretion of salt followed by water. All right, so this is receptor guanylyl cyclases. Okay, so many receptors are tyrosine kinases. And so, so here are the abbreviations. You've seen some of these. There are different kinds of domains. Yeah, they share different kinds of domains. So insulin is actually a dimeric receptor, right? There are 2 alpha and 4 beta subunits on the outside. And there are two subunits, tyrosine kinase domain subunits on the inside. Now you remember our discussion of VEGF receptor. R stands for the receptor. Anybody remembers what the VEGF is? Vascular.
Epithermal growth factor.
Vascular endothelial growth factor. Yes. So remember, this is a type of receptor where scientists are trying to develop drugs to suppress the growth of blood vessels, which are driven by tumors, right? Tumors trying to build their own blood vessels to deliver oxygen and nutrients for themselves. Now there is this platelet-derived growth factor receptor. Just different kinds of cells will express this one. Also growth factor receptor. Now this is epidermal. So you said epidermal growth factor receptor. Oh, I don't remember this one. This is fibroblast growth factor receptor. So all of these are growth factor receptors present on different kinds of cells. And all of these have external outside portion where the ligands bind, where the growth factors bind. And all of these have receptor tyrosine kinase portion inside the cell, which initiates phosphorylation cascades. And keep in mind that mutations are prevalent in these kind of receptors. Oftentimes, these receptors become oncogenic when there is no communication between these, right? No communication, and somehow this, when the communication is stopped, then they stop talking to each other, and the receptor tyrosine kinase is constantly on. And the cell constantly divides without any breaks. So the breaks are removed. And that gives rise to oncogenesis, right? The process of the tumor formation and growth. And so each of these receptors are actually involved in drug discovery, and there are many molecules now on the market that actually block the tyrosine kinase activity of these receptors. Some of them actually have very potent activity. So I told you about an antibody that Avastin that works against VEGF. Now there are small molecules that work against epidermal growth factor receptor. They're actually quite successful in the treatment of lung cancer. But not every single lung cancer patient, right? So a lung cancer patient whose lung cancer is specifically linked to the defect in the epidermal growth factor receptor, right? So first the lung cancer patient has to be, a biopsy has to be taken, and it has to be established that indeed these receptors are overexpressed in the cancer tissue of this patient. And if that's the case, then this patient can be successfully treated with these drugs. Okay, so we'll talk about insulin. So insulin, we know that it's a peptide hormone that's produced by the beta cells. Can never remember how to pronounce this. Every time I learn, I keep forgetting. Langerhans, or Langerhans in the pancreas. So once the insulin is produced, it reaches the target cells, such as liver, muscle, or fat tissue via the bloodstream. Binding of insulin to the insulin receptor initiates a cascade of events leading to increased glucose uptake and metabolism. And so if you have a problem with this system, you get diabetes. And there are two types of diabetes, right? And what's type 1 diabetes? Anybody remembers type 1? What is it caused by?
I think that's just regular diet.
No.
Type 1 is more genetic. It used to be called childhood diabetes because we don't, we only really saw it in kiddos, and they didn't generally live very long. We've gotten a lot better at treating it, obviously. And then type 2 is more, it can also be genetic, but it's more of diet and insulin resistance. Type 1, your body doesn't make enough insulin. Type 2 is insulin resistance.
Yeah. So type 1 is basically the immune system attacks and destroys these cells, right, in the pancreas. And type 2 is much more complicated. We still don't know the exact pathology of type 2 diabetes, probably on a level of insulin signaling inside the cell, but we still don't know very well. So so what does insulin receptor do? So when insulin binds to it, right? So there's this phosphorylation, autophosphorylation, which we'll talk about in a minute. But the point is that the how does it actually lead towards the uptake of glucose? So in a cell, there are these endosomes. Endosomes, basically large vesicles, which keep GLUT transporters in them, right? So GLUT transporters, they're anchored into these vesicles through their transmembrane hydrophobic domains. And then these endosomes, once there's a signal from the insulin receptor that insulin has arrived, they will merge and fuse with the cytoplasmic membrane and allow for the glucose to come in. Allow for the glucose to come in until insulin levels drop. The signal stopped, and then the cell membrane starts the process of this invagination like this, and buds off into these small vesicles, which then fuse with each other to form larger endosomes. And the glucose transporter will then just wait for the next meal, when the next portion of glucose arrives and ready to fuse with the cell membrane again. So, so this is glucose import in myocytes, for example. Myocytes are cells of what tissue?
Muscle cells?
Muscle cells. All right. So here is the complicated insulin signaling system. Very complicated. So obviously I don't want you to memorize this, or would you like to memorize this? Would you like this to be on the test? No?
No, thanks.
If you want.
Not particularly.
If you like to memorize these little letters, these abbreviations, some people like to memorize these abbreviations. So maybe you want to, and let me know.
If it is the only slide on the test, I will do it.
All right. So I told you that insulin receptor has two parts, and each part of the dimer has a tyrosine kinase activity as well as free tyrosines that can be phosphorylated. And so this part phosphorylates that part, and this part phosphorylates that part. And so you have a whole bunch of phosphotyrosines on the insulin receptor. That creates a binding site for IRS1. So IRS1, this is the only one I will tell you the abbreviation, unless you can tell me what it stands for. Do you know what IRS1 stands for? Internal Revenue Service. Anyway, just kidding. That stands for insulin receptor substrate. Insulin receptor substrate, which binds to phosphotyrosines and itself undergoes phosphorylation by the insulin receptor. And that is then recognized, the phosphotyrosine sites are recognized by adapter proteins known as GRIB2. That is recognized by SOS, Sons of the Sevenless. I know that one. Son of the Sevenless, which then binds to RAS. And RAS, as I remember, is a membrane anchored protein, right? Remember the specific anchor when we talked about protein anchored to cell membranes. And RAS then activates a kinase known as RAF1. RAF1 then phosphorylates MEK. Phosphorylates MEK. Now phospho-MEK then by itself is a kinase, phosphorylates ERK. Phosphorylates ERK. Phospho-ERK then enters the cell membrane and activates transcription factors, which results in gene expression and leads to the production of messenger RNA and the protein.
So this one goes into the nucleus?
Yeah.
Is that what's going on?
Mm-hmm. So, so for example, so basically the genes which are controlled by insulin receptor will be transcribed. So for example, this one, for example, glucose transporter GLUT4, right? So this is the, since there is insulin, there's lots of glucose, we need more GLUT4. All right, and yeah, that's good. Now there is also crosstalk between tyrosine kinase receptor and GPCR, which is an example of integration of signals. So in general, insulin signaling works, and the signaling through adrenergic receptor are opposing each other. Just think about it. So adrenergic receptor is a fight or flight receptor, right? You want this receptor leads to mobilization of resources. Now, insulin is a more type of let's store the energy, right? So there's also glucose in the blood, but we don't need this glucose. We will wait until we actually need to run. And let me take all this glucose, put it into the cell, convert it to glycogen or convert it to fats in adipocytes and so on and so forth. And so, so if there's lots of insulin, so there's a crosstalk. So the insulin receptor will, once the insulin is bound, then can phosphorylate. These are tyrosine. Remember why are tyrosines? Phosphorylate tyrosine residues. And also through the action of IRS1 and protein kinase B, phosphorylate serine residues on the adrenergic receptor. And once phosphorylated, adrenergic receptor undergoes internalization. Internalization, so we don't need to fight, we don't need to flight, we don't need these receptors right now. We need to actually store energy, right? And at the same time, insulin receptor will initiate signaling through its own way. So this is known as a MAP kinase receptor, sorry, MAP kinase pathway. MAP kinase, just in case if you ever hear this. So I think this is pronounced as SHIK. SHIK, GRIB, SOS, RAS, RAF, MEK, ERK, and then altered protein reception, altered gene expression. And so obviously all these signals coming in, there are also other receptor tyrosine kinases. The cell will then perform integration. All these, depending on particular metabolites, their concentration rises in some cases, it drops in other cases. The voltage across the cell membrane rises in some cases if there are ions involved, drops in some cases. And so overall response will be based on a crosstalk and integration of all these signals. All right, so yeah, this is the picture we already saw. I think the reason for the slide is just the phosphorylated tyrosine residues are generally bound by protein known as that have SH2 domains. SH2 domains So most proteins that bind to phosphorylated tyrosine residues will have these SH2 domains, which are designed to recognize phosphorylated tyrosines. So it's a highly conserved sequence of amino acids, which is shared by many proteins that bind to phosphotyrosines. And here's the, for example, a picture, right? So this will be the protein that has phosphotyrosine, and this is the SH2 domain shown here. You can see there's some hydrophobic cavity. There's a cavity that can accommodate the phosphate. So specifically designed to recognize phosphotyrosines. All right, gated ion channels. So gated ion channels. So there are two kinds of those. Well, there are the two major types are those that respond to changes in the membrane potential. So I told you that there is more positive ions on the outside and more negative on the inside, right, of the membrane. So there is a membrane potential, and they're also ligand. And so each of these, so the membrane potential, the voltage, and the ligand can bind to specific receptor sites and specifically to ion channels. And they can open or close the ion channel. So some of the important in the nervous system, voltage-gated sodium channels, nicotinic acetylcholine receptor, inotropic glutamate receptor, and gamma aminobutyric acid receptor A. So remember that there is the potential is between 50 and 70 millivolts. And that comes from the activity of sodium potassium ATPase, which pumps 3 sodium out and 2 potassium in, creating this potential. All right, so here's sodium potassium ATPase. So ATP undergoes dephosphorylation to ADP, and the energy is used to pump 3 sodium ions out and 2 potassium ions in, which creates the positive charge on the outside, like that, and also creates negative charge on the inside. And so if you obviously just don't do anything, right, and just have the channels, ion channels, what will happen? So these will be passive diffusion, not passive, facilitated diffusion, right? Without any energy, just ion channels. So sodium will come back in because it's being pumped out there. There is tremendous concentration gradient. It wants to get in. Calcium, remember, I told you the calcium concentration inside the cell is always low. So calcium wants to come in as well. Now, the interesting thing, so potassium also wants to get out because its concentration inside the cell is higher than its concentration on the outside of the cell. But the chloride is very interesting. Its concentration on the outside is very high. And yet, it still wants to get out. Why do you think that is? So if you simply go by the concentration, then the chloride would want to come in, not to get out.
Isn't it because of the electrochemical gradient? Like it's more negative on the inside of the cell, so it wants to flow towards the positive
Yes.
side of the membrane
yes.
outside? Okay.
Yes, correctly. So that's why you said electrochemical gradient. There are two components to that. There's electric and there's chemical gradient, right? So chemical is concentration, electrical is charge. And so even though its concentration is higher on the outside, but it still wants to get out because of the membrane polarization, right? It wants to depolarize the membrane by quenching these positive charges on the outside. All right, so let's see how the nerve signaling works. Now that we know that where these ions want to move. And so obviously nerve signals propagate. It is electrical impulses. And the propagation involves opening of voltage-gated sodium channels. Now, another point to keep in mind, that eventually results in calcium channel opening and release of neurotransmitter acetylcholine. And acetylcholine opens the ligand-gated ion channel on the receiving cell. So let's look at this picture. So here's our polarized membrane, plus on the outside, minus. So this is the axon of the presynaptic neuron. This is the synapse, also known as synaptic cleft. And this is a postsynaptic neuron. This would be similar to, no, never mind. So this is a neuron junction. All right, so the membrane is polarized, but the action potential is moving along. And action potential, what it involves is depolarization. So there are these sodium channels which are voltage gated. So as depolarization arrives with the action potential, channels open up, allowing for sodium to come in. Sodium comes in, right? Sodium comes in, and that you can see depolarizes the membrane. As it depolarizes the membrane, more sodium comes in and so on and so forth. So it's a wave, kind of a wave of sodium ion openings, sodium ion channel openings, which is brought about by depolarization of the membrane. So it's a wave of depolarization. And immediately following that is the repolarization. So potassium, remember, potassium wants to get out. And so since its concentration inside the cell is very high, and as the positive charge gets out, the membrane potential is reinstated. So it's back to where it came from, ready for the next action potential, right? So as one action potential is moving down the axon, the membrane is immediately repolarized, ready for the next action potential. So until it reaches the synapse where the acetylcholine vesicles storing acetylcholine are kept. And here we have lots of calcium, voltage-gated calcium channels. So depolarization opens calcium channels. The rise in calcium concentration will make the vesicles fuse with the axon cell membrane, allowing for the acetylcholine to be released into the synaptic cleft. So now there's lots of acetylcholine here, which then serves as a ligand for the ligand-gated ion channels. And so these allow sodium and calcium inside of the postsynaptic neuron. And again, as these come in, this starts a new action potential involving depolarization, moving down the postsynaptic neuron. And immediately following that will be repolarization involving the moving out of the potassium channels, potassium ions through the potassium channels. So obviously, so there are two types of you can see two types of ion channels. There's voltage gated involved, and there is ligand gated ion channels involved. Both are involved in nerve signaling. And so obviously, as far as the neurotransmitters, you can have different ones. So neurotransmitters add another point of diversity, in making brain very complex organ, right? Because all these neuron junctions also differ in the type of neurotransmitter which is released into the synaptic cleft. So this one is acetylcholine. Can you name any other neurotransmitters? What other neurotransmitters do we know?
GABA.
GABA. What does GABA stand for?
Gamma, anergic. It gets blurry after there.
So gamma aminobutyric acid. Anything else?
Serotonin.
Serotonin, good. Yep. Serotonin, GABA. Anything else? Surely you know this one, that people with Parkinson's don't have enough of it. What's the people with Parkinson's? What's the cause of Parkinson's disease? It's the lack of the neurotransmitter dopamine. Dopamine, because the neurons which are responsible for the production of dopamine are for some reason not functional in the brain. All right. So I think for integrins, we're just going to have one very short slide. So these are transmembrane proteins which link the cytoskeleton with extracellular matrix. So we talked about that. And there is some signaling there involved as well. So extracellular matrix can change its constituents from collagen, fibrinogen, fibronectin, and others, which can trigger skeleton rearrangement and gene expression. So there's quite a bit of interaction between cell and the extracellular matrix, and that is orchestrated by integrins. And finally, I want to mention the steroidal hormones, for example, right? Those that are derived from cholesterol. So you can see that actually now the steroids, they are not water soluble, right? Because they're so hydrophobic. And so they're brought by proteins that bind them in the bloodstream, and they're released into the cell. They cross the cell membrane, right? It's right here. They cross the nuclear membrane, and here they will bind into a nuclear receptor. Nuclear receptor will then associate itself with the hormone responsive element in the DNA, okay? Hormone responsive element, and the transcription of the new gene will start. And remember, the way transcription works is through this enzyme, RNA polymerase. RNA polymerase will start producing new messenger RNA, gene transcription, messenger RNA, translation on the ribosomes, new protein, and altered cell function. Any questions about this? All right. Well, I think we are at the end of what I wanted to tell you. There are other slides in this chapter, but I think we covered all the major ways for the signal to be transduced inside the cell. And I don't think we really need any more information at this point. As I mentioned to you, it's just a descriptive chapter for us. And so what did we learn? So we learned about receptors, right, which sense extracellular environment. They are specific to ligands. They often amplify the signal, can be modular. In modular, remember all these phosphorylation, dephosphorylation, and so on and so forth. And they desensitize to prevent excess signal. We just saw that. And so which ones are those? So GPCRs, they bind G proteins. Receptor tyrosine kinase is the second major one. Receptor guanylyl cyclases. Talked about voltage gain gated ion channels. And of course, dysregulation of intracellular signaling can lead to cancer. So like I told you, right? So let's say we're going back to this complicated picture of signal transduction. Now think about if RAF protein, for example, right? If it's defective and the signal between RAS and RAF is not transmitted properly, and RAF is constantly active. RAF is constantly active, and then MEC will undergo continuous phosphorylation regardless of what RAS is telling us, right? Because the interaction between RAS and RAF is disrupted. And so that can lead to cancer. All right, any questions about today's lecture? So I will send you a reminder about the review session for Friday. And as I mentioned to you, if you want to send me your questions before that, if you can't attend the Friday review session, I'll try to address those. And I'll be here. I'll become available. And
I'm sorry.
Yeah, go ahead.
Sorry to interrupt. What time is the review session going to be on Friday?
It's going to be 9 o'clock. So I will send a reminder.
Thank you very much.
Is the test next class, or is?
Yeah, the test is on December 2nd, which is on Wednesday. Yes.
And so the review on Friday, is that just for the final?
Yes.
Oh, thank you.
Yeah, the test on Wednesday, day after tomorrow, is going to be exactly the same as test 2. Nothing new. All right. Well, if you think of something, email me. If not, I will see you on Friday, because on Wednesday you're going to be taking your test. Sounds good?
Sounds good. Have a good day.
All right. Bye, guys.
Thank you.
Bye.
Thank you very much.
Mm-hmm.